Today's video would be studying oogenesis, and here we have the outline of all we'll be discussing. We'll be taking a look at the first meiotic division, the second meiotic division, and the clinical correlates. Now, oogenesis simply refers to the process and the steps by which ovum is produced and ovum develops. Okay, now of course, oogenesis takes place in females and it starts in prenatal or fetal life, and we'll be taking a look at this in the first meiotic division. Now, in a developing embryo, we have a bunch of cells. Okay, now these cells are precursors. Some of these cells are precursors for the cells that they would later develop into. An example of a precursor cell in the embryo is the primordial cell or the primordial germ cells. Now, the primordial germ cells are undifferentiated cells. They are stem cells and they are the precursor cells that would later on develop into sperm or ovum in the male and female respectively, as the case may be. Now, in the developing female embryo, these primordial germ cells arise at the yolk sac. Okay, now the yolk sac is a temporary structure that provides nourishment in the early embryonic life. Okay, now these primordial germ cells arise at the yolk sac. However, the yolk sac is away from the developing gonads, so these primordial cells would have to migrate. Okay, so they would have to migrate from the yolk sac down to the developing gonads, and since we're considering oogenesis, the developing gonads would be considering here the developing ovaries. Okay, so these primordial germ cells would have to migrate to the developing ovaries of the embryo, and in this case we're considering a female embryo. When they migrate to the ovary, of course all of this is taking place in fetal or prenatal life. When the primordial germ cells get to the ovary, they differentiate. Okay, so they differentiate into oogonia. Now these oogonia cells multiply. Okay, so they multiply, they proliferate by mitosis. Okay, the word used is proliferation, and it's simply a fancy word for multiplication. The oogonia multiplies or it proliferates in the ovary, in the developing ovary of the embryo by mitosis. So first we had the primordial germ cells arising from the yolk sac. Okay, the endoderm of the yolk sac. Endoderm of yolk sac. And then these primordial germ cells, because they are at a location away from the developing ovary, they would have to migrate. Okay, so they migrate to the developing ovary, and when they migrate, they differentiate. Okay, so differentiation takes place, and these primordial germ cells differentiate into oogonia. Now these oogonia cells proliferate or multiply by mitosis. Now we have more oogonia cells. Okay, so more oogonia. Now this oogonia further differentiates. Okay, so it further differentiates and further develops into primary oocytes. Now it's good for us to take note of the fact that the oogonia is diploid. Okay, so it has the chromosome number of 46 for the human, and of course it's X. Also, the primary oocyte is diploid. It has total complete number of chromosomes, and it's also an XX. Also important for us to take note of the fact that when referring to the chromosomes, we could have two sister chromatids as we have here. So we could have two sister chromatids forming a chromosome, but also one chromatid as this. Just one chromatid can also be referred to as a chromosome. Okay, so a chromosome could be two sister chromatids, and it could also be just one chromatid forming the chromosome. Okay, however, in the oogonia as well as in the primary oocyte, what we have is a two sister chromatid chromosomes. Okay, so we have 46 of this in the oogonia and in the primary oocyte, and XX because this is in a female. Okay, of these 46 chromosomes, we have 22 autosomes. Okay, so the autosomes are somatic chromosomes. These somatic chromosomes are chromosomes that carry genetic information for somatic cells, which are body cells. So the general body cells are the somatic cells, and the chromosome for these somatic cells are called the autosomes. And then we have a pair of allosomes. Okay, so the allosomes are also sometimes called the sex chromosomes. The sex chromosomes, in the female we have the XX, and in the male we have the XY. However, of course, because we are referring to a female here, the oogonia is an XX. It has an XX allosome. Now we have the primary oocyte, which are diploid cells. Okay, so this is diploid. Now, this primary oocyte undergo the first meiotic division. Now, this first meiotic division is not completed in fetal life. In fetal life, it stops at the prophase 1. Just to visualize what happens, we'll take the primary oocyte, this to be the primary oocyte, and we have 46 chromosomes, but we'll take two chromosomes representing this 46 chromosomes. Okay, so we have 46 chromosomes here represented by two chromosomes. Okay, and since the 46 chromosomes form 23 pairs, we have just, in this case, our representation would have one pair of chromosomes, or let's use 4 chromosomes and 2 pair of chromosomes. So we have something like this. Now these chromosomes are made of two sister chromatids. Now what happens in the prophase 1 is that this first of all, the chromatin granules that we have actually condense, and so we can now see, so probably initially we had something like this, just the chromatin granules scattered all around. In prophase, these granules condense, so we can now see the chromatin, we can now see the chromosomes as they are here, and the two sister chromatids are also evident. Now what happens is that homologous chromosomes come together and form a bivalent. So a homologous chromosome, for example, we could have this and this coming together to form the bivalent, and of course this and this coming together to form the bivalent. Now it's good for us to take note of the fact that crossing over takes place in this phase. So we have what we call crossing over. Now initially, these chromosomes, we have those from the maternal side and from the paternal side. Okay, so we have this chromosome, say from the maternal side, and another chromosome here from the paternal side. Now what happens in crossing over is that the chromosomal arms literally cross over. So we could have something like this. Okay, could have something like this with the chromosomal arms crossing over and forming a chiasma. Now when these homologous chromosomes separate in anaphase, they take away a part of each other. So we could have this taking a part. Okay, so the blue arm, a part of the blue is now stuck to the red, and for the blue we have here, a part of the red is now left on it. Now this crossing over is what allows for genetic combination and variation in general. So this is an importance of meiotic division, as we would see. Okay, so this is what happens in prophase. Now it's important for us to also take note of the fact that when the cell enters the resting phase at prophase 1, so the first meiotic division stops at prophase 1 in fetal life up until puberty, where we have these pre-ovulatory primary oocytes that would now continue the first meiotic division because of an increase in the luteinizing hormone level. But it's good for us just for now to take note of this fact that the primary oocyte stops. So we have the primary oocyte. It stops the first meiotic division at prophase 1. Prophase 1. Taking a look at the picture that we have here, we can see first we have the primary oocyte, then we have the primary follicles. The primary follicles are cuboidal in nature, so cuboidal epithelium, so to say, and as we would see, these primary follicles would mature and they would later form the ovarian follicle that help in the production of estrogen. We can see the next step where we have the growing follicles of this primary oocyte. The follicles continue to grow, and then at this stage we now have the zona pellucida. The zona pellucida is an amorphous glycoprotein layer on the oocyte. Okay, as we can see in the picture here. Also fluid begin to form within this follicular cell layer, and then this fluid coalesces. Okay, it comes together and then forms the antrium. So the antrium is this fluid filled space that we have here, and also we have the outer cuboidal epithelium here for also forming part of the Graafian or the ovarian follicle. Now let's go further into still looking at the first meiotic division. Now we have our primary oocytes that have stopped the first meiotic division at prophase 1. Okay, and of course now we have the baby. The baby is born at this point. The female baby is born at this point, and the female child already has all of these oocytes. The female child already has all of the oocytes that she would need throughout her lifetime. This newborn baby, which we have a fancy word for neonate. So we have this female neonate that is born with about 2 million primary oocytes. Okay, all the oocytes that she would need for throughout her lifespan. Now only about 400 are actually ovulated. Okay, only about 400 of these oocytes mature into only about 400 of these primary oocytes mature into secondary oocytes and are ovulated. Now, all of these primary oocytes, remember, they are at prophase 1 and they remain at prophase 1 just before menarche. Okay, just before menarche and just before puberty hits. Now, in the pre-ovulatory phase, this primary oocytes in the pre-ovulatory phase, when there is an increase in the luteinizing hormone, now the luteinizing hormone is a hormone secreted by the anterior pituitary gland. So the hypothalamus releases gonadotropin releasing hormones, gonadotropin releasing hormones. Now this hormone acts on the anterior pituitary. We have the anterior pituitary and the posterior pituitary, but for this video we'll be considering the anterior pituitary gland. Now this gonadotropin releasing hormone acts on the anterior pituitary gland, and taking a look at the name, it's gonadotropin releasing hormone. So this hormone stimulates the release of gonadotropin. Okay, now the gonadotropins are the LH and the FSH. Okay, this is short for luteinizing hormone, and the follicle stimulating hormone. Now these hormones are released, and as we would later see, the follicle stimulating hormone stimulates the development of the ovarian follicles for the release of estrogen, while the luteinizing hormone trigger ovulation and the development of the corpus luteum, as well as the production of progesterone. So as I said earlier, we have this newborn baby with all of the primary oocytes. So this newborn baby grows up, and in the pre-ovulatory phase, into a teenager. Newborn baby grows up into a teenager, and in the pre-ovulatory phase there's a surge in the luteinizing hormone. Okay, so there's an increase in the luteinizing hormone, and this increase in the luteinizing hormone kickstarts the meiotic division again. Okay, so it kickstarts the meiotic division. Remember that the first meiotic division stopped at prophase 1. So the luteinizing hormone kickstarts it, and then from prophase we have metaphase. Okay, so from prophase 1 to metaphase 1. Okay, so now we have our cells. Remember, the cells stopped at a bivalent, so we had bivalent homologous chromosomes like this. Now what happens at metaphase is that these homologous chromosomes come together. Of course, we have our spindle fiber has already been formed. Okay, so spindle fiber has already been formed. These homologous chromosomes come together and they line up at the equatorial plane. Okay, so we have the equatorial plane. They line up at the equatorial plane, and we have the spindle fiber just attached to these chromosomes in this way. Okay, spindle fiber, another spindle fiber. This is what happens in metaphase, the arrangement of the chromosomes along the equatorial plane, and this is sometimes called the metaphase plate. Okay, so we have the metaphase plate. So just as a recap, first we have the primordial germ cells, okay, migrating to the ovary. When they get to the ovary, they differentiate into oogonia. This oogonia proliferates or multiplies by mitosis, and it increases in number. Okay, the oogonia is further differentiated into primary oocytes. Now, so far we have been dealing with diploid cell. The primary oocyte is a diploid cell, meaning that it has the total of 46 chromosomes. Okay, now we have this primary oocyte, and this primary oocyte begins the first meiotic division, but it stops at prophase 1. Okay, it stops at prophase 1, and all of this takes place in the fetal or prenatal life. Now we're born, the baby is born, the female baby, the female child is born, and this female child has all the oocytes that she would need throughout her lifetime. She's born with about 2 million primary oocytes. Some of these primary oocytes regress. Only about 400 actually develop into secondary oocytes and are ovulated. Okay, now what we are looking at is how after during puberty this primary oocyte develops. Okay, so we said in the pre-ovulatory phase at puberty before menarche. Okay, so this is before menarche. What happens is that there's an increase in the luteinizing hormone, and this increase in the luteinizing hormone triggers the start. Okay, it triggers the start of, or rather it triggers the continuation of the first meiotic division. So initially the primary oocyte was just resting at this first prophase 1, but due to the increase in this luteinizing hormone, then it kickstarts the process, and then now we are at metaphase. Now, as I stated earlier, at metaphase we have these cells lining up at the equatorial plane. Okay, so it actually looks something like this. Okay, so they line up at the equatorial plane, something like this, and then like this. Okay, so we have the spindle fiber attached to the chromosomes. Okay, now the next step of meiosis is anaphase. Okay, and what happens in anaphase is that we have this spindle fiber retracting. Okay, so the spindle fiber retracts, and as it retracts, it pulls the chromosomes towards opposite sides. Okay, so it pulls the chromosomes towards the opposite poles. Okay, so we have the cell, the primary oocyte, something like this, and then the spindle fiber pulls the chromosome towards opposite poles, something like this. Okay, towards opposite poles, and the spindle fiber continues to retract. Okay, now of course the next step in meiosis, so this is anaphase 1. Okay, the next step in meiosis is telophase and then cytokinesis. Now what happens is when these chromosomes get to opposite poles, we might expect that okay, the cell would divide into 2 equal daughter cells. However, this is not so. The division is actually unequal, and this division leads to the formation of a secondary oocyte. So we have a secondary oocyte and a polar body. Okay, so the secondary oocyte takes the most part of the cytoplasm. So we have something like this. This could be the secondary oocyte, and then the polar body is just very small. It only takes a very small portion of the cytoplasm. The first polar body takes only a small portion of the cytoplasm, and it later disintegrates. Okay, so it later disintegrates while the secondary oocyte has the ovum forming capacity, and it's the secondary oocyte formed here that would go on to form the ovum. So at this stage, the first meiotic division is completed. This secondary oocyte begins the second meiotic division. Okay, it begins the second meiotic division, and the second meiotic division proceeds into the prophase 1, and or rather the prophase 2 and the metaphase 2. However, the secondary oocyte stops at metaphase 2, and at metaphase 2 it is released into the uterine tube. Okay, so at metaphase 2 the ovary releases this secondary oocyte into the uterine tube. Now if fertilization takes place, then the ovum proceeds the second meiotic division down to anaphase and telophase and cytokinesis. Then it fuses with the pronucleus of the sperm. However, if fertilization does not take place, the ovum or the secondary oocyte at metaphase 2 disintegrates within 24 hours. Okay, so it's good for us to take note of this. So far we have seen that when the newborn baby is giving birth to, she already has all of the primary oocytes, and all of these primary oocytes are at prophase 1. Okay, in the resting phase of prophase 1, stops at prophase 1. Now the clinical correlate we'd be considering is Down syndrome. Now Down syndrome is also known as trisomy 21. It's known as trisomy 21 because Down syndrome occurs as a result of the presence of three chromosome 21. Okay, so generally in a normal embryo would have just two chromosome 21, one from the paternal side and one from the maternal side. However, trisomy 21 occurs when there are three of this chromosome 21. Now how this results, this usually results in pregnancies at a much older age. Okay, because of the prolonged nature of the prophase 1. Take for example, you have a woman that gets pregnant at an older age, maybe at an age very close to menopause, so maybe at an age of 40. So take for instance, you have the lady, for example, this is just an instance, maybe at an age of 40, and of course ovulation takes place. Now the ovulation starts as a result of the increase in the luteinizing hormone. Okay, so we have the primary oocyte, the crossing over here, and at anaphase, what we saw here, we saw that the homologous chromosomes would then retract to either side of the cell. Now what happens in Down syndrome is what we call a non-disjunction. Now in a non-disjunction, it occurs when the homologous chromosomes that form the bivalent fail to separate at anaphase. Okay, so when they fail to separate at anaphase at this stage, what we have is, take for instance, we have something that looks like this. Okay, so we could have this homologous pair of chromosomes that do not separate. Okay, so they move to the opposite pole of the cell, and then we have this pair that separates. This will result in a cell that looks something like this. Okay, and another like this, and then we have this that looks like this. So this would result in the cell formation that looks like this. Now if this happens to be the secondary oocyte, what happens is that now we have a non-disjunction. This is similar to having 24 chromosomes in a particular gamete and say 22 in the polar body. Now when this secondary oocyte with 24 chromosome is fertilized by a sperm with 23 chromosomes, it will result in the formation of a zygote with 47 chromosomes, which is abnormal. Okay, now when this is as a result of an extra chromosome 21, okay, so the chromosomes each have numbers. So when the chromosome that exhibits the non-disjunction is chromosome 21, it leads to trisomy 21, and trisomy 21 results in the formation of an embryo that would later be born with Down syndrome. If the secondary oocyte is the one that gets 22 chromosomes while the polar body gets the 24, when this secondary oocyte with 22 chromosome is fertilized with a sperm containing 23 chromosomes, it results in an embryo with 45 chromosomes. Okay, and this is known as monosomy. Usually embryos with monosomy do not go further in development, so it could just lead to the death of the embryo. As stated earlier, this condition may arise as a result of pregnancy at a much older age. Okay, the end of all the processes we have considered so far, we have the oocyte released in this form, the secondary oocyte. We have the first polar body here, okay, and then the nucleus, the zona pellucida, which is an amorphous layer around the ovum or around the secondary oocytes and it's made of glycoprotein. And then we have the corona radiata, which is a multiple layer of cells. Also, we can see here that we only have the first polar body. It's important for us to take note of the fact that the second polar body is formed only when fertilization takes place. Now, going by our outline for this video, we have come to the end of the video. If you liked this video, please give it a thumbs up and don't forget to subscribe. Thank you for watching till the end. Bye.